Okay, we're picking back up with what is inside the cell. So let's go through what's inside the cell. We've mentioned it before, but there are ribosomes inside the cell. And then there are the membrane-bound organelles. And just to review, the membrane-bound organelles are structures contained by a membrane that are a specialized component of a cell, meaning they do a specific job. And this is great because eukaryotic cells are very large. And the larger a cell gets, the more efficient it has to be in order to actually exist in any functional way. So organelles provide that efficiency that a larger cell requires. So let's take a little bit more detail about ribosomes. They look kind of like dots when we're drawing them. And they're found in the cytoplasm or attached to organelle membranes. And the main important, huge detail about ribosomes is that they are the site of protein synthesis. Proteins are made at the ribosomes. So basically what you have is that in the nucleus, the gene for a protein is copied into a message with that protein's recipe. And then that recipe travels out of the nucleus into the cytoplasm of the cell, where it meets up with a ribosome. And at that point, amino acids are brought to that ribosome, and the protein is put together according to the recipe in the message. That is the basics of ribosomes. And we'll talk a lot more detail about it much later on. But for now, that's what you need to know about ribosomes. So they're made of two chunks, actually. They're creatively named the large subunit and the small subunit. As you can see, the large subunit is large, whereas the small subunit is small. Amazing. Anyway, here it is all put together. I'll go ahead and give you a couple of seconds to draw this. Yep. Got it? Finish drawing it? Okay, good. No, I'm not going to put that on an exam. Anyway, here we're going to continue on to the organelles because they're so important. So you can see there's a large variety of organelles in a eukaryotic cell. So let's talk about them specifically. We have vesicles, the nucleus, the rough endoplasmic reticulum, or the rough ER, the smooth endoplasmic reticulum, or the smooth ER, the Golgi apparatus, the lysosomes, the vacuoles, then the mitochondria and chloroplasts, and that is our list for 105. So, not a ton, 1, 2, 3, 4, 5, 6, 7, 8 organelles to know. So, I don't expect this to be a huge problem for you. All right, so organelles are part of what we call the endomembrane system. Not all organelles, but most organelles are part of the endomembrane system. We call it the endomembrane system basically because it means that all these organelles are participating in sort of a continuous membrane. They're all connected in some way. So, what is part of that endomembrane system? Well, we have the nucleus, the nuclear envelope, the endoplasmic reticulum, both rough and smooth, the vesicles, the Golgi, and the lysosomes, and vacuoles. So, a large variety of things. All right, so what is the point of having the endomembrane system? Well, again, functional. All these different things are connected together, which means all their jobs are related. Essentially, the endomembrane system is involved in building proteins, transporting proteins throughout and outside of the cell, making energy, moving lipids, detoxifying poisons, all of this. So the endomembrane system has all these things linked together to make all these functions more efficient so that the different ingredients can get to the right spot more easily because all the membranes for these organelles are essentially linked. So let's talk about the very first organelle I've mentioned, the vesicles. These are an important part of the endomembrane system, and they're just an important part of being the cell. A vesicle is a small bit of membrane that buds off of a structure. And as it buds out, it forms sort of a bubble. And when it disconnects, the bubble closes, and essentially this vesicle allows the cell to transport substances into the cell, within the cell, and out of the cell. Most organelles can produce vesicles. So here is a cell membrane, right? And it's starting to bud. And this is what I meant by budding. These dark areas, these are chunks of molecules that need to be passed around inside the cell. And so it buds further out. You can see that bud starts to close, right, into a bubble. And then it will eventually pinch free here and disconnect and be a self-contained bubble. And then when this vesicle, say we're traveling from one organelle to another. So for instance, we're traveling from the rough endoplasmic reticulum here. So this is the rough endoplasmic reticulum, and this is the membrane for the rough ER. And we form our vesicle and pinch it off, and it needs to travel to the Golgi apparatus. So how is it going to take its contents that it carried from the inside of the rough ER and give them to the Golgi apparatus? Well, let's just look at this picture and review. So here we had the rough ER, put its contents into the little bud, which budded out, closed, and disconnected into a vesicle. And the vesicle floats over to the Golgi apparatus, which is also a membrane-bound organelle. Because it has a membrane, the vesicle's membrane and the Golgi's membrane can fuse. And so they meet up and start to fuse, and then it forms this bud here, and then it does this, and we eject the contents into the Golgi. Same exact process, just in reverse. So let's take a look at vesicle movement in a nerve cell. So this is pretty neat. So these are vesicles that have been stained to be more visible. So it is pretty neat that you are actually seeing vesicle movement. So there we go. I really like this video. It's a great example of the types of things you can actually see in microscopy with the correct stain. All right, moving on to the nucleus. The nucleus is surrounded by the nuclear envelope. The nuclear envelope is a double membrane that has pores running through it. Now, when you look inside the nucleus, you will see a dark spot inside the nucleus. That is the nucleolus. However, it's important to note, very important to note, the nucleolus is not an organelle. It is not an organelle. The nucleolus is a very dark spot because it happens to be a very thick clump of DNA. Imagine the DNA inside the nucleus kind of like a ball of yarn. Some of it is loosely sort of scattered in there, sort of long, loose loops, but you've got a ton of it, so you need to make it compact and easily stored, so you get to a point where you start to ball up that DNA, ball up the yarn, and you get this nice compact ball of yarn that's very dark. That's the nucleolus. It's dark, but it's just DNA. That's the site where DNA is most concentrated. So this is our membrane, and here you can see, if you follow my mouse pointer, right, here is my double membrane. So you can see an outside layer and an inside layer. That's the double membrane. And here's a good example of it connecting up. This is the nuclear envelope connecting to the rough endoplasmic reticulum. So that's that endomembrane system, how they're continuous. Here are the little pores. You can see this is an excellent spot right here. You can see the pores allow the passage of things across the entire nuclear envelope. And then here's all my DNA, sort of loose bits here, And then here's the really dark concentrated DNA that makes the nucleolus. Talk a little bit more about DNA. DNA is, of course, inside the nucleus. You normally find DNA in this sort of loose, willy-nilly unwoven form called chromatin, loose, uncoiled DNA fibers. And we can contrast chromatin with chromosomes. In case you've ever heard the term chromosomes, chromosomes are when you take the chromatin and twist and coil and fold it up into this highly concentrated structure. And it ends up looking a little bit like an X. So just kind of neat. One chromatin fiber condenses into one chromosome. So when you look at this picture, this is all DNA in the form of chromatin. We'll talk more about chromosomes when we talk about mitosis and meiosis. All right, next up is the endoplasmic reticulum. The rough endoplasmic reticulum is sort of a folded membrane system, and it has ribosomes embedded all over it. In fact, it is called the rough ER because under a powerful microscope, when you see the rough ER, it looks like it would be rough to the touch, and that's because it has all of those little ribosomes studded all over those folded membranes. So it looks rough. That's literally the only reason it's called the rough ER. So if it has thousands of ribosomes embedded on it, well, what is it involved in? What are ribosomes involved in? That's right, protein synthesis. The rough ER is all sorts of involved in protein synthesis. Now, the smooth ER has no ribosomes. That's why they call it the smooth ER. Doesn't look rough. Not having ribosomes, it's not so much involved in protein synthesis. Rather, it can be involved in synthesis of steroid hormones like testosterone and estrogen and progesterone, detoxifications of drugs and poisons, especially in liver cells. So here's a rough ER. You can see all those ribosomes studded all over it. Looks like if I went to touch it, it would feel all scratchy. It would feel rough endoplasmic reticulum. And then here's the smooth endoplasmic reticulum with no embedded ribosomes. And you can see here, this, when we say it's a folded membrane system, you can see all these little folds in the membrane here. That is what they mean when they call the ER a folded membrane system. It's made of a large number of little folds. And here again you can see nuclear envelope continuous with the rough ER. Then over here you can see the rough ER continuous with the smooth ER. We're all endomembrane system here, continuous membrane. All right, so we move on to the next organelle on the list, the Golgi apparatus. The Golgi apparatus is a funky organelle. Products from the endoplasmic reticula, whether it's smooth or rough, are sent to the Golgi. In fact, it's vesicles that bud off of the endoplasmic reticulum that go to the Golgi apparatus. Whereas here, the nuclear envelope is directly connected to the endoplasmic reticulum. The endoplasmic reticula, smooth and rough, are connected to the Golgi apparatus via vesicles. Now, because vesicles are forming from this membrane and connecting into the Golgi, we're still having sort of continuous membranes. So, the ER puts its products, whether it be a protein or a steroid, into a vesicle and sends it over to the Golgi. The vesicle fuses with the Golgi and ejects its product into the Golgi apparatus. And then the products are modified, stored, and then sent to other destinations, whether it needs to be excreted out of the cell, whether it needs to be sent to another part of the cell. The Golgi is responsible for getting it to where it needs to go. It's kind of like the warehouse, right? The ER here, these are kind of like factories where you make a product And then once you make the product at the factory, it's sent to the warehouse. And the warehouse stores it, or it modifies it with the shipping labels, and then it's mailed out from the warehouse. That's what the Golgi does. So, like I said, ER makes a product, Vesicle carries the product from the ER to the Golgi, and then it's modified. How is it modified? Little phosphate tags indicate where in the cell the product has to go. So that's what we mean when we say modifying. It's just little phosphate molecules that get bound to the surface of that compound, whatever that product is. And they help the cell move that molecule, that compound, whatever it is, to where it needs to go. And so then they leave the Golgi through another vesicle. And then they go to other organelles, or they go to the plasma membrane for secretion. So here's a Golgi membrane, or here's a Golgi apparatus right here. Again, big folded membrane here. This is actually a microscopy picture. And then this is a three-dimensional representation. These little blue vesicles are vesicles that are arriving at the Golgi apparatus, bringing those products. And then they move through the Golgi and they're modified, and then they're sent out via other vesicles to where they need to go. So let's talk about secretion. So when a cell moves a product outside of the plasma membrane, that's secretion. So how do products leave the cell? Well, a vesicle. So vesicles can fuse with the plasma membrane, and when it fuses, it becomes part of the plasma membrane, and anything that was inside that vesicle is ejected on the other side of the plasma membrane. So it releases its contents outside of the cell. Here we go. Here's my rough ER making a product. Well, actually, this is the smooth ER. This is the rough ER. So a product is made, and it goes to the Golgi, and then it's put into a vesicle, which moves out to the plasma membrane. And you can see here's the vesicle fusing with the plasma membrane, and everything's leaving the vesicle. We're secreting something. Next up is the lysosome. The lysosome can be thought of sort of like the stomach of a cell. The lysosome is how a cell digests large molecules. So a lysosome is kind of like a vesicle in that it's sort of a little membrane sac, except that a vesicle can carry practically anything, whereas a lysosome has a very specific payload. Inside a lysosome, you find digestive enzymes that help break down certain compounds, and then you also find an acidic fluid. So between the acid and the enzymes, you can break down larger compounds. So, like I said, sac similar to a vesicle, contains enzymes and an acidic fluid, and then the enzymes break down and digest materials. My great example for lysosome action, during embryonic development, our hands are actually webbed. There's skin between all of the fingers. But when we're born, we have individual fingers. It is lysosomes that are actually responsible for breaking down all of the skin between the fingers. Kind of neat. So what do lysosomes digest? External macromolecules brought into the cell. So, for instance, the cell membrane gulps food into food vacuoles or vesicles, depending on the size. If it's something that's really big, it's in a food vacuole. If it's something that's relatively small, it's in a vesicle. That's literally, it's just a naming thing. Big nutrient molecules, big, large macromolecules go into food vacuoles. Smaller molecules, small macromolecules end up in vesicles. Internal components are also broken down and digested by the lysosomes, because that allows you to break down those internal components into little recyclable bits. So external macromolecules are broken down into monomers, and then internal components are recycled. So any organelle or other component that breaks down or gets old, you can break it down into something else and then make a new one. So these are food particles here. They're relatively large. So when the cell takes them up, it's very big, so we're going to call it a food vacuole. And here's my lysosomes binding to it, and they fuse with the food vacuole because, well, food vacuole is a membrane-bound sac. Lysosomes have a membrane. The membranes can fuse. Any organelles that have membranes can fuse. So the lysosomes fuse to the food vacuole and release their enzymes and acid and break it down, and then any crap that's left over is kicked back out of the cell. All right, next up is the vacuole. There are different kinds of vacuoles. Vacuoles are kind of interesting. There's the food vacuole, which I mentioned is just a very big vesicle. So we give it a different name because science just wants to make things difficult on you. Then there's the contractile vacuole. The contractile vacuole actually has a completely different purpose. That purpose is being a bilge pump. A contractile vacuole pushes water out of a cell. Remember when we talked about osmosis? What happens when you put a cell in a hypotonic environment? Which direction does water go? Did you say into the cell? I hope you did. That's the direction water goes when a cell is put in a hypotonic environment. Well, if a cell happens to have a contractile vacuole, it basically collects water and then pumps it out of the cell. And that can help the cell combat the influx of water from being in a hypotonic environment and keep the cell alive, keep the cell from swelling up and bursting. And finally, we have a central vacuole, which you see in plants. So this is a contractile vacuole right here, and this is a little single-celled protista organism, a paramecium. And he lives in fresh water, so it's not entirely impossible that he'd wind up in a situation where he's in hypotonic conditions. So instead of swelling up and exploding, these contractile vacuoles start to push water out of the cell. So the food vacuole brings food into the cell, and it fuses with a lysosome. Let's take a look. So this is kind of neat. This is a paramecium, a type of little single-celled organism. This is a tube to the outside, and it uses little hairs and beats them to pull fluid into that tube, and then it forms food vacuoles at the tip of the tube. You can watch that happen. So we're filling it up, we're filling it up, we're filling it up, and when it gets full enough, it's going to just disconnect from there, and we can start making another food vacuole. So ready? Just about there, I expect. There it is. A nice food vacuole filled up. And now there's another one filling up. So pretty neat. And then there's the contractile vacuole, as seen in freshwater protists, and that pumps excess water out of the cell. And let's take a look. Okay, so here are our little protists, and let's zoom in. And that's a contractile vacuole right there. You can see those weird little star shapes they had in the drawing earlier are not all that, far off from what you see. Here's the contractile vacuoles functioning. And each time they pump, they're pushing water out of the cell so that it doesn't swell up. So that's just super neat. Oh, there we go. And see, there's those little star shapes there. So, see, there they go again. Really, really cool. Then finally, there's the central vacuole that you see in mature plant cells. So in older plant cells, you get a bunch of different vacuoles nearing each other, and they decide to form a little club, and they end up fusing together. And all these small vesicles fuse up, and they form a really, really large central vacuole. So there you go. That's why you have the name central vacuole. It's a big vacuole in the center of the cell. Its whole purpose is to store inorganic ions like potassium and chlorine. It can also store organic nutrients for when the plant isn't getting enough. It can also store water when necessary. It can even store poisons. Why would you want to store poisons in the plant? Did you say defense? That's right. Even plants have defensive capabilities. In this case, there are plants that store poisons in their central vacuoles. And those poisons can damage or at least repel herbivores that want to eat the plants. So, kind of a cool little arms race going on between plants and animals. Plants develop defenses against animals, and then animals adapt to those defenses. It's just very cool. All right, so I'll come back talking about the power plant of the cell. We'll call this good for now because I am sure I'm probably right around or over 20 minutes.